Block 2, Unit 4, Drosophila Development and Cell Cycle Checkpoints. Fruit flies undergo 3 stages of development before emerging as adults: egg, larva, and pupa. At room temperature, fruit flies can develop into adults within 1 to 2 weeks. The egg and larval stages span approximately 8 days, while the pupal stage lasts 3 to 5 days. The adult fruit fly lives for several weeks in the lab. Fruit flies undergo 3 stages of development before emerging as adults: egg, larva, and pupae. At room temperature, fruit flies can develop into adults within 1 to 2 weeks. The egg and larval stages span approximately 8 days, while the pupal stage lasts 3 to 5 days. The adult fruit fly lives for several weeks in the lab. Female flies are most fertile in the first 4 days after eclosing, or emerging from the pupal case. Once impregnated, female fruit flies are capable of laying more than 500 eggs. Eggs are usually laid in fruit or other sugary, decaying organic material. Fruit flies choose which breeding site in order to ensure a food source for their larvae after they have hatched, as well as protection from certain predatory species. Optimal temperatures for fly eggs range from 75 to 80 degrees Fahrenheit. What do fruit fly eggs look like? Fruit fly eggs measure only 1/2 millimeter in length. Under a microscope, they are yellow in color and appear to be the shape of a grain of rice. 24 hours after a female fruit fly lays her eggs, larvae hatch. These larvae ultimately mature into adults that feed, breed, and lay their own generation of eggs. Larvae are the small worm-like early stages of fruit flies. These larvae prefer the wet fermenting areas near fruit or other sweet items. They can also be found commercially in baking areas where sweet fruit fillings may have fallen and have been moistened with wash water. Larvae prefer fermenting items, and if the fruit or other source becomes too well fermented, the larvae will no longer feed, as fungi and bacteria may become too prevalent. After fruit fly eggs hatch, larvae begin to feed on the decaying material within which they were laid. Larvae consume as much food as possible in order to store energy and nutrients for the upcoming pupal stage. Fruit fly larvae undergo moulting stages known as instars, during which the head, mouth, cuticle, spiracles, and hooks are shed. During the larva's third instar, it crawls to a drier area to pupate. The pupal case is formed from the larval skin as it darkens and develops a hard surface. Inside the pupa, larvae begin to develop into adult flies. The parts of the fly are derived from sacs of cells called imaginal discs. The pupa, therefore, serves as a type of cocoon. An imaginal disc is a sac-like epithelial structure found inside the larva of insects that undergoes metamorphosis. Once the larva turns into a pupa, almost all the larval tissues degenerate and the imaginal discs turn into the external structures of the head, thorax, limbs, and genitalia. The rest of the adult external structures, abdomen, originate from nests of histoblast cells that are formed in the larva. The reason imaginal discs are of such interest to developmental biologists is that they provide a powerful tool to study multiple aspects of development and biological mechanisms underlying disease. They are one of the reasons that make the fruit fly Drosophila melanogaster, which from now on we'll simply call fly, an excellent model organism. Why are they called imaginal discs? Imago is a Latin word that means image and is used by entomologists to refer to the mature stages of insects. The imaginal discs get their name since they are disc-shaped structures that will originate from the imago. How many imaginal discs does the fly have? There are 6 imaginal discs in the fruit fly. The epidermis of the head, thorax, and limbs of the fly come from 10 bilateral pairs of discs containing cells. Their names are genital, eye, antenna, these will form the head, wing, haltere, legs, obviously they are the precursors of the 6 legs, and the genital disc. Where do the discs come from? Imaginal discs form as groups of 10 to 50 cells that segregate from the epidermis of the embryo. Most of these primordia invaginate and remain connected to the larval epidermis only by a thin primordial stalk. When metamorphosis is complete, the adult fruit fly pushes its way through the anterior end of the pupa. This process is called eclosion. One could say that the adult fruit fly ecloses from the pupal case. Initially, the fruit fly is light in coloration with expanded wings and an elongated abdomen. Within a few hours, the fruit fly darkens, extends its wings, and expands its abdomen. Approximately 48 hours after emerging from the pupa, female fruit flies are sexually mature and can begin breeding and laying eggs. Adult fruit flies are fertile for the entirety of their lifespan. Female fruit flies can store sperm from multiple inseminations for use in future egg production. How long do fruit flies live? The average natural lifespan of fruit fly adults in optimal temperatures is 40 to 50 days. Female fruit flies are capable of mating and laying several batches of eggs in that time, allowing the fruit fly population in a home, for example, to multiply quickly. The lifespan of the fruit fly is heavily influenced by temperature. The cell cycle is an ordered series of events leading to replication of cells. The cell cycle is divided into 4 major phases. In cycling or replicating somatic cells, chromosomes are replicated during the S synthesis phase. After progressing through the G2 phase, cells begin the complicated process of mitosis, also called the M phase, which is divided into several stages. Chromosomes condense during the prophase period of mitosis by tightly folding loops of the chromatin fiber attached to the chromosome scaffold. Sister chromatids, produced by DNA replication during the S phase, remain attached to the centromere and multiple points along their length and become aligned in the center of the cell during metaphase. During the anaphase portion of mitosis, sister chromatids separate and move to opposite poles of the mitotic apparatus, or spindle, segregating one of the 2 sister chromatids to each daughter cell. In most cells from higher eukaryotes, the nuclear envelope breaks down into multiple small vesicles early in mitosis and reforms around the segregated chromosomes as they decondense during telophase, the last mitotic stage. The physical division of the cytoplasm, called cytokinesis, then yields 2 daughter cells. The Golgi complex and endoplasmic reticulum also vesiculate during mitosis and reform in the 2 daughter cells after cell division. In yeast and other fungi, the nuclear envelope does not break down. In these organisms, the mitotic spindle forms within the nuclear envelope, which then pinches off, forming 2 nuclei at the time of cytokinesis. Following mitosis, cycling cells enter the G1 phase. The period before DNA synthesis is reinitiated in the S phase. Postmitotic cells in multicellular organisms can exit the cell cycle and remain for days, weeks, or in some cases, like in nerve cells and cells of the eye lens, even the lifetime of the organism without proliferating further. Most postmitotic cells in vertebrates exit the cell cycle in G1, entering a phase called G0. G0 cells returning to the cell cycle enter into the S phase. This re-entry is regulated, thereby providing control of cell proliferation. Regulated protein phosphorylation and degradation control passage through the cell cycle. The complex macromolecular events of the eukaryotic cell cycle are regulated by a small number of heterodimeric protein kinases. Protein kinases are enzymes that add phosphate groups to other proteins to change their activity. The concentrations of the regulatory subunits of these kinases, called cyclins, increase and decrease in phase with the cell cycle. Their catalytic subunits are called cyclin-dependent kinases, or CDKs, because they have no kinase activity unless they're associated with cyclin. Each CDK catalytic subunit can associate with several different cyclins, and the associated cyclins determine which proteins are phosphorylated by the CDK cyclin complex. Passage through three critical cell cycle transitions, G1 to S phase, metaphase to anaphase, and anaphase to telophase in cytokinesis, is irreversible because these transitions are triggered by the regulated degradation of proteins, an irreversible process. As a consequence, cells are forced to traverse the cell cycle in one direction only. The G1 checkpoint. In higher organisms, control of the cell cycle is achieved primarily by regulating the synthesis and activity of G1 CDK complexes. These complexes are at the G1S point of the cell cycle. Extracellular growth factors, called mitogens, induce the production of G1 CDK complexes. The activity of these and other CDK complexes is regulated by phosphorylation at specific inhibitory and activating sites in the catalytic subunit. Once mitogens have acted for a sufficient period, the cell cycle continues through mitosis, even when they are removed. The point in late G1 where passage through the cell cycle becomes independent of mitogens is called the restriction point. A different molecular mechanism is used to control cell cycle progression through the G2 checkpoint, which prevents entry into mitosis in the presence of unreplicated or damaged DNA. Arrest of the cell cycle at this checkpoint is mediated by a protein kinase called checkpoint kinase 1, or CHEK1, which is activated in response to DNA damage or incomplete replication. CHEK1 phosphorylates the protein phosphatase CDC25, thereby preventing CDC25 from dephosphorylating and activating CDC2. In the absence of CDC2 activation, progression to mitosis is blocked, and the cell cycle remains arrested in G2. Described differently, when there is DNA damage, CHEK1 adds a phosphate group to the phosphatase CDC2. Remember that a phosphatase is a protein that removes a phosphate group from another protein. When there is a phosphate group on CDC2, it is inactivated and does not remove phosphates. What is checkpoint kinase 1, or CHEK1? CHEK1 is a multifunctional protein that helps regulate the DNA damage response and participates in regulating progression through the cell cycle at S, G2M, and M. The specificity of CHEK1 is conferred by phosphorylation of different sites on the protein. In other words, CHEK1 does different things depending on where a phosphate group is added to the CHEK1 protein. It is a kinase that phosphorylates or adds a phosphate group to another protein called CDC25. For our purposes, we'll focus on the role of CHEK1 in the G2M transition, but it's important to keep in mind that CHEK1 is involved in many different signaling pathways related to DNA damage and cell cycle progression. You will not be responsible for knowing the specifics of these interactions, but know that they exist. When is CHEK1 activated? CHEK1 is activated when there is DNA damage, and the DNA damage response, or DDR, is activated. The DNA damage response is a complicated network of signaling pathways that lead to temporary arrest of mitosis so DNA repair can take place or apoptosis can be initiated, depending on the extent of the DNA damage. More specifically, CHEK1 detects the presence of single and double-stranded breaks in the DNA. When DNA damage occurs, two proteins can be activated, ATR or ATM. Each can in turn activate CHEK1, depending on the cell cycle and the organism. What is the effect of CHEK1 activation? Once activated, CHEK1 phosphorylates CDC25 proteins, also called CDC25A in humans. Upon phosphorylation, CDC25A is inactivated. In its active state, not phosphorylated, CDC25 induces progression into mitosis. Without active CDC25A, the cell cycle is stalled between G2 and M, so DNA can be repaired. So what if CHEK1 is missing? In the research performed in the Discovery Lab, you may have the opportunity to work with flies that have the checkpoint gene grapes in Drosophila mutated. The mutation is referred to as GRP1 in Drosophila. The mutation causes no protein to be made from the gene. This mutation was identified in Drosophila by Ethan Baer and colleagues at University of California, San Francisco in 1991. By using a genetic technique, the research group generated 3,768 lines of mutant flies. They identified mutations that were lethal when homozygous. The mutant called grapes earned its name because it caused the nuclei in larvae to look like clusters of grapes. The nuclei form clusters in embryos from grape-1 The nuclei form clusters in embryos from grape-1 mothers because the nuclei fail to separate during telophase, but only in embryos that have gone through 12 nuclear divisions. The nuclei at this stage have migrated to the cortex of the embryo. This is called syncytial division, where the nuclei divide, but cytokinesis hasn't taken place yet. At that time, the gene sequence was not known. It wasn't until 1997 that another group further characterized grape-1 mutant flies. They observed that when the embryos from grapes' mutant mothers were irradiated, there was a large increase in the number of single and double-stranded DNA breaks compared to wild-type larvae. They hypothesized that the grapes protein was involved in DNA repair and cell cycle progression. It was known that the mutation was caused by a genetic insertion, so the group mapped the insertion and identified the nucleotide sequence of grapes. Sequence analysis revealed an open reading frame that encoded a protein that was 513 amino acids long. The sequence was very similar to a serine threonine kinase called CHEK1. It was therefore concluded that the grapes gene was a homolog to CHEK1. CHEK1 in cancer. When functionally normal, CHEK1 responds to DNA damage by arresting the cell cycle and allowing DNA repair. Because of its role in regulating progression of the cell cycles, it should not be surprising that if CHEK1 is not functioning normally, cell division can become dysregulated. For example, if there's too much CHEK1 in cells, overexpression, cells are more easily transformed into tumor cells. This has been found in breast, colon, liver, gastric, and nasopharyngeal cancers. It is thought that perhaps extra CHEK1 allows cells to respond more quickly to damage in rapidly dividing cells. CHEK1 also plays a role in tumor resistance to therapy. If CHEK1 activation is abnormally high, tumor cells can be resistant to several different types of therapy, including radiation or drug chemotherapies. This is observed in many types of cancer, including glioblastoma, prostate, and lung cancers. Conversely, if CHEK1 protein is missing or has decreased activity, the cell will not be able to arrest the cell cycle to repair DNA damage caused by some drug chemotherapies and radiation. Therefore, inhibitors of CHEK1 are in development as a chemotherapy for their ability to cause cells to accumulate DNA damage and die. To review the pathway as a whole, it's easiest to start at the bottom, where the cell makes a decision to either arrest or proceed with the cell cycle. At number 1, under normal circumstances, CDK cyclin is dephosphorylated by the phosphatase CDC25A, causing progression of the cell cycle. At number 2, when DNA damage occurs, single or double strand breaks can occur, activating ATM, ATR. At number 3, you can see that upon activation, ATR phosphorylates the kinase CHEK1. At number 4, you can see that activated CHEK1 phosphorylates CDC25A, which marks the protein for degradation. Without active CDC25A, the cell cycle is arrested.